Good morning, ladies and gentlemen. I'm Upul Sanak, and my co-chair is Dr. Harsa Satish Chandra. And on the stage will be Professor Kalari Krishna, who's an associate professor of biomedical informatics at Mayo Clinic and a lead computational biologist at the Center of Individualized Medicine. Professor Kalari Krishna.
So here are the learning objectives for today. But before I begin, I was just wondering, to just know my audience better, I was wondering how many of you know the term bioinformatics, multi-omics, precision medicine, and artificial intelligence. Can you please raise your hand? Okay, excellent. So I'm going to give some basics, kind of introduce you these terms first, and then talk about the omics footprint. It's a method that we recently developed and just got that published last month. And then I will talk about the application of that. And then also talk about the interpretable AI models, because AI is known as a black box, right? So it's kind of hard to interpret it as what people think, but there are ways to do that. So I will talk about that. How did we develop those interpretable AI models? And then comparison of our method that we built, the omics footprint, with the rest of the state-of-the-art methods. I will kind of show those results as well. So what is bioinformatics? Bioinformatics is an interdisciplinary field that includes engineering, computer science, chemistry, biochemistry, biology, statistics, mathematics, data science, and everything, right? It is a combination of fields where that can be used to used to investigate biological data, like clinical data, research, or any other data sets, a complex, high throughput data sets, basically. So this term bioinformatics kind of emerged in 1970, but then kind of became more popular in 1990s when they began this project, Human Genome Project. And then in 2003 is when they first completed the first Human Genome Project. After that, there were a number of studies like genome-wide association studies where DNA, the genomic data was measured and then the phenotype was associated. I'm not going to go through each year what happened, but I just want to say that the momentum kind of picked up more after the Human Genome Project got released in 2003. And then where we are right now, in 2024, it's a lot more about the multi-omics, deep learning AI, and precision medicine. And when I say multi-omics, I want to make it it's not just genomics. It is different types of omics. And that's what I will explain in my next few slides. What is multi-omics, what is AI, and what is precision medicine? So multi-omics, the DNA, for example, here is the sequence of ACGTs. Right? Whole genome sequencing, when people talk about it, is basically you have the patient sample and you're trying to see what kind of these sequences exist in that patient's gene, right? So ACGT is a combination of those letters. And when it is an RNA, it is a transcription of that DNA, and where you can measure the gene expression. So individual genes, we have a lot of genes, about 30 to 40,000 genes. We have gene expression determined for each of those genes, which means a person could have a set of genes highly expressed and low expressed as well. So it's just a quantitative number. And then the third is a protein where that particular three sequence, like the alphabets, GAG, for example, becomes a glutamine, right? An amino acid. It just gets converted. This is basic biology. I'm sure all of you know. I'm just trying to remind you all. And then there's metabolomics, there's lipidomics, there's microbiome. There's so many different types of omics there, epigenomics and others. So all these is what we derive that as multi-omics. So integration of multi-omics data. Here, for example, for a patient, you have different types of omics data, genomics, epigenomics, and these are all different types, right? But I want you to focus on this particular array here, for example. It is like a patient's genes are measured, say 30,000 genes are measured in the transcriptomics. So on this like in your Excel file, like on your x-axis, you have the genes, and on your y-axis, you have the patients, right? So individual patients. So say, for example, I have my rows as the genes, and then the columns as the 100 patients that we are interested in. So you have this in a tabular format. So different types of data are in different tabular formats, right? So what normally we try to do in bioinformatics is we try to take each type of omics data and then try to associate that with the phenotype, kind of correlation or associations. And then say, okay, these particular biomarkers are associated with this phenotype. So you could do that individually and then say, okay, these are the genes that are associated, or these are the proteins that are associated individually. But can we do it in a multi-omics fashion where you're able to integrate all these omics data types? Because at the end of the day, they're measured on the same patient, right? Or on the same individual. You have just different types of data. Like, for example, in the clinical data, you have your age, BMI, and like other measures from the same patient, like the imaging data and everything else from the same patient, right? It is the same thing here. It's the different types of data, omics data, but measured on the different omics data measured on the same individual. So multi-omics data is nothing but it's just a multi-omics, as the name says. And then can we integrate that is what we were trying to look at a couple of years ago. There are some benefits. Obviously, we can improve the diagnostic precision because, say, with the clinical data, you're able to explain 60 to 70% of the variance of the disease because of the heterogeneity. But then you add this omics data, then you're able to increase your, performance, like the prediction itself, right? So we can also identify the novel targets, and we can also understand the disease heterogeneity better. So one example, I'm just talking about any complex diseases, as long as you have omics data, and it could be multi-omics or individual omics too, because it is hard to get multi-omics data on all patients. But if you have such data and then and you're able to correlate that different types of that data with the phenotype, then you're able to identify biomarkers and others. And it could be done for any cancer or like diabetes or neurological disorders or others as well. I hope by now I was able to explain you what a multi-omics concept is. Now I will kind of try to explain the AI, deep learning, and ImageNet. There's nothing to fear about these terms. These are pretty simple terms, but just like, let me explain you. AI, for example, is like human-like intelligence. It performs tasks like language understanding, image recognition, and all, right? Just like I see someone and I'm able to recognize who they are, right? That's an image that's stored in my database, which is my brain. So that's the deep learning is like a human brain. It's a neural network of, it's a part of AI that has neural networks in it. And it's like brain where the cells are like here, the nodes, and we are able to recognize the complex patterns. The ImageNet is like my database, for example, where I'm able to save the images of each of you and then maybe able to recognize you later on, right? So that's the same thing. ImageNet is a database where there's 14 million images that are curated manually and stored there. So when you see, like when an AI sees a strawberry, for example, because it has seen so many strawberry images, it's able to recognize it. Similarly, a dog or a cat or anything, right? Because it's been trained. It's just like a medical new intern. When they come in, they don't know a lot about it, right? But when they see a lot of these cases and then learn a lot about the case and the images, they are able to identify and predict the next patient who comes in and say, okay, this is what must be happening, right? It's the same thing. ImageNet is a database of like these images of the data. In this case, it's visual images of like strawberries, blueberries, whatever. And then ImageNet, the database, is used to identify, like our brain, how we are using it, right? To identify, that's the same network, like a brain in the neural network that it's using. And then the artificial intelligence is like just a human-like intelligence that we are applying. So it's pretty simple. it's just a terminology that we all have to get used to in future. And again, an example of AI, I'm sure you all know about this study, the diabetic retinopathy study that happened I think several years ago, a decade ago, with 10,000 patients. After feeding the images of the 10,000 patients, it was able to recognize the patients with the cases and controls very clearly. And how that happened is because of this ImageNet. Because obviously you don't have 14 million images of the diabetic retinopathy patients, right? You only have a small set of it, but that's what the ImageNet is for. ImageNet is where you use the transfer learning. So the images that are there, you use that and then start using it for your own projects. So you combine the knowledge from the ImageNet and your own study or the images that you have, and then start looking into the predictions. Now, I hope I can kind of talked about a little bit about AI and explained you that concept. Now I will kind of introduce you to the precision medicine concept. In this case, I'm going to just use a cancer as an example. We have a patient population. And then in this case, the standard approach is give them a paclitaxel or some drug. And then only 20% of them it works because obviously, they have different types of mutations and different clinical and other characteristics, right? So how can we do this better, the tailored approach? How can we do that better? So basically, if you take these different patients here, different colors or different subtypes, if you think that way, if you're able to differentiate their subtypes nicely and then able to put them into different treatment buckets, that's what is precision medicine. And at NCI, it's a clinical NCI match clinical trial that's happening right now, where 6,000 patients are entered into this trial, and then about hundreds of genes are sequenced. Like their mutations are identified, and then based on their mutations and clinical data, they're put into different buckets. So for example, if they have PIK3CA mutation in AKT, something going on, they put it into this purple bucket. So it's called kind of like a bucket trial. So this is happening right now. And with that, I think I introduced all the three concepts now, and I will move on to kind of introduce the omics footprint itself. So if I take all my abstracts that I've published, 140 or more papers, and then take all that and put it into a word cloud, again, we are data scientists, so we love doing these kind of fun things. So if you take all the abstracts and then put it into a word cloud, you can clearly see I focus a lot in cancer, gene expression, bioinformatics, Alzheimer's, and several other things. And here is what we do in my program. We have majorly, we have divided our methods that we develop, because I'm more a biomedical engineer by training. So I'm interested more in development of methods. So we have these 6 major ones, but today I will focus on the omics footprint and explain you what it means. This is a paper that got published on 24 October 2024 here with an impact factor of 16.5. I'm going to kind of describe that now. Okay, since I gave you all the basics, now let me explain you. I told you about the tabular format. Remember that? I was telling, x-axis will have the genes, your rows as your genes and your columns as your patients, right? So if you wanted to represent the omics data for an individual patient, right? So you could do this, because at the end of the day, each of these genes are associated to the chromosome and chromosomal position, right? So we all have 22 chromosomes, and X and Y are XX, right? So those are the chromosomes. So we were thinking at that time, a couple of years ago, this was a very novel idea, because we were thinking at the end of the day, everything is annotated to genes or chromosomal positions, instead of having them in individual tables and how can we make it better? So we thought we'll build omics footprints. For example, what I meant by that is going from outside to inside. This could be one type of omics data. So this could be gene expression data. And the bar here represents high expression and low expression. Green is low and normal expression as well. right? And then the copy number data or like mutations, fusions, any kind of data, proteins. I didn't show the protein data here, but yeah, you can introduce the protein data, methylation or anything. It is just at the end of the day, a circle. So for an individual, it is a footprint of that individual's multi-omics data as one image. So all the tabular data that we have is converted into an image for an individual. Again, I want to say this is not for multi-individuals, but for an individual, you could do this, okay? And I'll explain you how we can do better after this. So right now, for storing the tabular data for one patient or an individual, it takes about 15 megabytes of data. With this kind of omics footprint, you can save that in 100 KB. It's amazing, like you're able to condense all that data into an image. There will be some loss of information, but not a whole lot. And then have this into a picture so that we are able to put it into our electronic medical records. That's the hope, right? At the end of the day, you have all the clinical data, but you do this sequencing and the data is all over in different people with different stats teams or bioinformatics teams or other teams. Can we make those into a footprint so that we can put it back into the electronic medical records? So with that, like this is like, for example, a 700 patient data. And you can see all these are the different types of footprints from those 700 individuals. So now we're going to start mining these images instead of like the tabular data. So once these are converted into images, now it's like, I can have multiple, as I said, the different circles are different types of omics data, and I could use them for multiple purposes: classification, biomarker identification, or drug response prediction, or risk survival, just like any other stats methods. But now I'm going to use images instead of tabular data. In order to do that, we had to build like a pipeline. And this is the whole workflow divided into 3 major stages. One is the first is like take the multi-omics data, convert them into images. The second is like build the AI model such that we are able to predict. Now, great, we are predicting it at 80%, 90% accuracy, but can I make this better? Because I know I'm able to predict these 2 subtypes that are available in that disease, but can I say what are the features that are differentiating these subtypes? Can I say that this combination of these PTEN mutation and then PI3KCA or something is differentiating this particular subtypes? Basically, it's like interpretable AI models. Can I do that is what we were trying to do and build this workflow. So here are some examples of like how we use the lung adenocarcinoma data, breast cancer data, and some the different types of breast cancer data as well, both the histologic and as well as molecular subtypes. And then we were trying to create these images and then try to test our pipeline that we have developed. We also did this for Alzheimer's disease, and we are also working on migraine and other data sets as well. Because this one is a pipeline which could be used for any type of disease. It's not, like it's not only for cancer, but in this case, I'm just going to show you a few cancer examples. Here, the first question we wanted to ask is like, there are multiple AI algorithms out there. Which one is the better one to make that as a default in our framework? So we compare different algorithms with different data sets. And then we said, okay, these are the different types of architectures that are there, AI architectures. And then efficient net V2 is the better one. And then we did that for the lung. For example, on the x-axis, you're seeing different types of architectures. On the y-axis, you're seeing the performance. And then you see this particular efficient net V2 in all these different cases is the one that's performing the best. So obviously, we made that as our default architecture. And then there's always this question like, it's hard to obtain multi-omics data on patients. Is one omics data type enough, or like do we need 2, 3 or more, right? So even we didn't know that, right? So we started investigating that by taking like just the gene expression alone and then a combination of 2 individual like omics data types and then 3 or more, and asking this question like which one would give us better performance? Obviously, we saw that multi-omics gave us better performance, but gene expression was good enough. And we did that with different lung and breast cancer examples as well. For the sake of time, I won't get into the details, but I'm just saying I just wanted to say that the multi-omics was doing much better than the other omics data types. Now I'm just going to walk you through this figure where it's a comparison of our omics footprint, which is at the beginning here, with two other imaging methods. And this is tabular methods, multi-omics integration tabular methods, supervised and unsupervised methods. So in bioinformatics field, you can't just say your method is good and here is my framework and I'm good. I just have to go and compare it with everything that's existing. So I was able to find out like 12 other methods, but they're not anywhere like imaging-based methods. They were more like tabular-based methods and others. And these two imaging ones had their own defaults because they can take more than 3 omics data types. So when we did the comparison across these different types of drugs, our method was performing the best. And then, as I said, interpretability of these models also we were able to show, like in this case, we were looking at the differences between the lobular and ductal carcinomas. And we saw that one type of multi-omics data was differentiating the lobular versus the ductal. And this is the first time ever, to our knowledge, someone has shown the use of multi-omics data in this fashion. We were also able to confirm the known findings. And there were one last slide here is like, this is talking about like, there is an, like people's belief, like we all believe that imaging, like for applying AI methods, you need a lot of images for training. So we have shown that like even 130 images are good enough as long as you use the right framework. Like I said, you have to use the ImageNet and others. And then in this case, even at the 130 images, we were falling down here. As you can see, the performance kind of goes down here. So 130 images is good enough, at least in the lung cancer case. And then the goal is obviously we have all the data in the bytes. Can we bring that to the bedside? And we are doing a lot of studies in single cell digital spatial profiling, developing a lot of methods. Now it's even beyond what I described, because for each single cell, you can measure multi-omics data. And then can we see the cell-cell interactions, and can we identify the biomarkers better? And then also integration of that with the whole slide images, like in cancer, we get the H&E slides and we're trying to integrate that with that. Multimodal integration will be what you'll see in future, where it'll be omics data, imaging, EMR, and clinical data. And we are heading in this direction. And hopefully in a couple of years, we'll have a paper out in that area as well. And thank you, Dr. Suranga Manilgama, for the invitation, and Nimalka for arranging all this for us, and all the SIMCON organizers. I also want to thank the funding agencies as well, and my whole team, my both clinical team as well as my own team members. Thank you all.
Thank you very much, Professor Kalari, for that fascinating lecture, showing us an extremely new field and what's happening in it. I'm sure the audience might want to ask one or two questions. Just a thought on what you said, Professor Kalari.
Oh.
Yes, may I just ask a question? Now, we're used to managing patients according to guidelines for any condition, and that's based on the phenotype. So, with precision medicine coming in and the machines learning fast, do you see a future where guidelines will be created by the machines for treatment of patients? Because we are going to are we going to replace the general guidelines that we make based on phenotypes?
I don't think that'll ever happen. It'll just be a second pair of eyes that will be complementary to the clinician, kind of helping them with the, instead of looking into databases and searching on your own, we are hopefully these algorithms will only help you better.
Okay, thank you.
Mm-hmm.
In the interest of time, I think we'll wind up the session, and may I invite Dr. Puldisanayaka to present the certificate of appreciation to Dr. Kalari.